There we go. Okay. Oh, cool. So, can everybody see? Yes? It's kind of just black and white here. Yes. Good. Cool. Thank you. So my name is Ben Busby. I do research at the National Center for Biotechnology Information. If that doesn't immediately ring a bell, PubMed is also out of there. I do not work for PubMed. I do research in comparative genomics for that organization. Today is my last day doing research in comparative genomics. As of Monday, I will be the genomics outreach coordinator for NCBI. So I will be doing a lot of talking to researchers about using NCBI databases more often, as well as submitting more sequence to NCBI. The other thing I do besides that is I chair the bioinformatics department for a graduate school on the NIH campus called FAES, or the Foundation for Advanced Education in the Sciences. We teach about 10 courses a year, about 4 of them in next generation sequencing, and the rest we teach scripting courses in Python, Perl, and R, as well as some medical genomics and personalized medicine courses. If you happen to find yourself in Bethesda on Monday, Wednesday, or Thursday nights this evening, we are teaching medical genomics and personalized medicine. On Mondays, we are teaching next generation sequencing on Wednesdays. That's a little bit more of an advanced class, and we are teaching introduction to Python on Thursdays. Actually, the guy who teaches Python drives down from multiple to teach Python. Very good.
Are those classes open to the public?
They are open to the public, absolutely. And not only that, I told the administration at the graduate school that I was coming to give this talk, and they gave me course calendars, like schedule. They have like little booklets. They are in my briefcase, so if you want one, if you happen to be in Bethesda at night, or know anybody else interested in science, that is, I would be happy to give you a booklet, because otherwise they will probably just sit in the back of my vehicle for another couple of months, at which point I will throw them away. Yes?
Streamed or captured?
What's that?
Are the classes, they have to be on premise or?
They are on premise as of right now. Once in a while, we will teach a next generation sequencing workshop remotely. Also, NCBI is doing a lot more video casting than it currently does. We'll start doing a lot more and having face-to-face webinars. So keep an eye out for that. And those things will be free through the National Center for Biotechnology Information. Additionally, on Udemy, we're planning to release a version of our next generation sequencing course, most likely sort of late spring, early summer this year. So if you're interested in next generation sequencing and how that works and how you can do data analysis, so on and so forth, that's something to check out. The nice thing about that, for something like DIY Bio, is there is a ton of data that you can access pays to have a lot of that data sequence.
So Dr. Busby, did I hear, I just want to make sure, did I hear the word free?
Absolutely. You heard. NCBI puts a lot of data up for free. Now, that said, there is data that is protected by HIPAA because there's personally identifiable information. So human genomic data with phenotypic information associated is not readily accessible, and other personally identifiable human data is not necessarily, it may be HIPAA protected, it is not necessarily accessible. If you are affiliated with an academic institution, you can apply to get access to that data, and actually the likelihood that you will get access is pretty high. 66% of applicants are approved, and the remaining 33% that are denied, apparently you're staying at the same, no, the 33% that are denied, a lot of them are patent trolls and such, looking for data. That said, I don't know, and I actually kind of doubt that sort of the DIY Bio group has been approved, but I think if you could show reasonable protections and so on and so forth, it's probably the case with me. But that's not what I'm here to talk about tonight. What I'm here to talk about tonight is personalized medicine, understanding genomics, epigenomics, and metagenomics. And this is really a talk built for non-scientists. How many of you are scientists professionally? Oh, great. Oh, wow. Okay, so 3 or 4. All right. So I have a slide. I have one slide that is for professional scientists. I believe that's slide number 29. So please, no questions from biologists until slide. I'm kidding. Please feel free to ask questions, but we'll try to hold it. So I'll talk about complex etiologies and etiological syndromes. Slide 29. That'll be for scientists, and the rest of you guys, if you don't understand this stuff. It's okay. If you don't understand anything else, please ask me. Are there any clinicians in the room? Great. All right. I always say things that offend clinicians. So that's good. All right. So next slide, please. All right. So what do people in the world die of? As far as I understand, this is data from 2010, and this is the world, not just the United States. We see that most people are dying of cardiovascular disease, obviously followed up by infectious and parasitic disease, and lots of people do die of cancer. That's something that's growing quite a bit in the developing world. And the rest of the things are spread out. Interestingly, only 0.3% of people die of war. So that indicates that we could theoretically, if we were interested in human life, change our national funding priorities some.
Hey Dave, can you focus that a little?
What's that?
Can you focus that a little?
We focus a little?
Or is it just my eyes?
It looks your eyes.
The phone is a little bit small.
Well,
You have to move
can we put the slides up later?
Absolutely.
Okay,
yeah.
good.
I will.
All right.
I will send you the slides. All right,
Cool.
next slide, please. So what goes into this vast majority of things people die of? Well, obviously things like toxics and nutrients, diet, viruses, bacteria, but also, of course, cellular factors like our genome and our epigenome, as well as our metagenome. And we're going to define this and talk about the bacteria and fungi that live on us and in us. And those all contribute to disease states. And I think it's important to remember that disease states also feed back into these things, and many of these things play into each other. So just to define some terms, next slide, please. So we can call these sort of as a group environmental factors. And then these, like I said, we can call cellular factors. These would be ecosystem factors. And once again, there's a widespread interplay feeding into disease states. Next slide, please. So this is a completely hypothetical example to illustrate what I think is the most important point of this talk, which is that singular etiology or singular genomic causes for a disease are typically fallacious. So the idea, we hand out these, the Platelet Center at NIH where I work, hands out these shirts that say, working together for the cure to breast cancer. And that the idea that there's a cure to breast cancer, or that it's even one disease, it's just a completely fallacious notion. I mean, there are diseases that, you typically have one major factor for, say, cystic fibrosis. 90% of cystic fibrosis cases are caused by one particular mutation. But the vast, vast majority of diseases are caused by hundreds of factors. And so let's take this hypothetical example. And for the purposes of this discussion, we're going to call it stomach disease X. And we're going to say that there are 400 causes of stomach disease X, and 100 of them are purely genomic. And we're also going to say that those 100 tend to happen early in life. And that's something we actually see borne out by data that at birth, 25 to say 30% of disease incidences are purely genomic. By the time people get to say the age of 65, it's somewhere more in the range of 4 to 10% of disease presentations are purely genomic, and we don't think have much to do with environmental factors. A lot of that's based on twin studies, as you can probably imagine. So say this is 100 cases, right here, next slide please. And so say you can look at just one type of case and narrow in on one genomic etiology. And I sort of phrase that hypothetically, but at this point, we can actually do that. And I'm going to show you some examples. Next slide, please. So, and getting to the point that it's absolutely false to say that we haven't made major healthcare discoveries with genomic information. But it's that small discoveries are made every single day. So maybe we find something about a cohort of 10 patients, or patients that comprise 1% of a particular disease cause. And we can't necessarily treat all of those people right away. But there are some cases where we can. Next slide, please. And a great example of that is maple syrup urine disease. So maple syrup urine disease is a horrendous disease that occurs in Amish populations. And basically, there's neurodegeneration that occurs between the ages of 2 and 4. And basically, normal, healthy kids end up becoming, end up in a vegetative state and stay that way for the rest of their lives. So there's a place in Lancaster County, a couple of physicians got together and built this thing called the Center for Special Children. And I want to point out that the Center for Special Children deals with all of the Amish kids in their community and attends to their health needs as well as the health needs of some of the parents there. And they have an annual operating budget of about $500,000 a year. They also do some genomic sequencing. They got some of these kids, they were able to sequence them. And what they were able to pull out was that there was a mutation in a gene which processes branched chain amino acids. So in this particular case, arginine. So what happens is arginine builds up in these kids' brains between the ages of 2 and 4 and causes this horrible neurodegeneration. So the great news is that if, between the ages of 2 and 4, you give them formula, which has lysine swapped out for arginine, the kids are totally fine. And they live totally normal lives for the rest of their lives. So now they've had dozens of patients around the country, which they've been able to cure of this horrible thing. Not to mention the human pain and suffering, but we're talking a price tag of something like $10 million per patient. That's saved by this clinic. That, in addition to all the other stuff they do, is able to do this for $500,000. Next slide, please. So, I just wanted to have you take a look. This is the way scientists might look at genomic variants, and this is actually a page in NCBI. And here, what we see are variations that have been reported in the literature to correlate to disease. Now, this gene is BRCA1. I'm sure most of you have heard of BRCA1, obviously. Angelina Jolie recently elected, maybe it was BRCA2. She recently elected to have surgery based on it. No mutation of BRCA2 gene. Sure enough, people with a deletion of this exon of BRCA1, they are very, very likely to develop breast cancer. If you look at breast cancer cell lines, often, but not always, remember, there's a genetic etiology. There are multiple genomic etiologies for breast cancer as well. But if they have this mutation, the cell lines are very likely to get breast cancer. So this is a very penetrant mutation. But what we're doing is we're able to build information about genomic variants for a lot of genes. Now, the question is, how does this translate to the clinic? Next slide, please. So, right now, and this is today, physicians can order panels of genetic tests, and they're relatively inexpensive. So you can order a combination of up to 1,500 genes from these people Invitae for $1,500. So it's relatively inexpensive to go from public data, as long as it's been verified, typically in multiple families, to be able to screen for this type of stuff. And in some cases, early detection is very, very helpful in dealing with some of these diseases. So, in terms of personalized medicine or precision medicine, when we deal with just genomics, the goal is not to cure cancer tomorrow or in the next year. The goal is to grab 2% of disease X and 2% of disease Y, and 2% of disease Z, and see what we can make treatable, then have physicians screen for it, treat these people early. And I'll say this at the end, but the real goal of this is really not to, we don't really, the big goal is not to prevent death. It's really to give people more healthy years. If we could do early detection of these kinds of things, and a bunch of the things I'm going to talk about later, we could do things like give people 25 more healthy years. That has a significant impact, obviously on the people's lives, but on our society and our economy as well. Next slide, please. So, another thing I want to talk a little bit about are toxic or infectious diseases in a genetically labeled background. So, this gets to the sort of interplay of genes and environment. What we're starting to see is that sometimes people have genomic mutations that don't present as anything immediately. But when they have a particular infection or are exposed to a particular compound, they may present with a disease. So let me show you a couple examples. Next slide, please. But first, one application. So one thing that most of you probably know or intuit or can think about, different people have different responses to pharmaceuticals. Sometimes pharmaceuticals are very effective in certain populations versus other populations, certain individuals versus other individuals, and then sometimes some pharmaceuticals have very nasty side effects in certain individuals versus other individuals or other groups. And this is big business. So Thomson Reuters and Numeni just launched a large groundbreaking initiative to repurpose drugs, to take drugs that do one thing in one context, and they can help with other things. And there are good examples of where drug repositioning has worked. So for example, antidepressants, things that act as antidepressants in certain populations with certain genomic contexts might act against certain kinds of cancers or neuroendocrine disorders in other populations. Does that make sense to everybody? Is everybody kind of with me? All right. So let me show you some specific examples of where people get sick from such things. So this is, this is data from NIEHS, which is the National Institute for Environmental Health. Anyway, so what they did I thought was very interesting. So they took several hundred individual cell lines from a bunch of what we call half-death populations. So these people are of Caucasian descent from Utah. These people are Great Britain, Japanese people, and African people. And what they did, this is just one compound out of about 150 compounds they tested, and they tested for an effective dose of 10%, which means they were looking for damage to those cells at a certain dose. And what they found with this particular compound, or what we found with this particular compound, I thought was particularly interesting, that there, in each of the, whatever this compound is, most people have what we might call sort of a normal response. There's probably a more or less normal distribution here, but we see that there are some people that are really outliers, and they're much, much more sensitive to this compound than others. That has actually been borne out over the last few decades. People working in particular kinds of chemical plants, we find people that are particularly sensitive to the chemicals in these chemical plants. In the next couple of years, we should be able to detect this at a large scale for large numbers of people. Now, so, as many of you know, recently the Genetic Non-Discrimination Act was passed. So, it makes it very difficult for employers, for better or for worse, it makes it very difficult for employers to screen people and say, hey, you probably shouldn't work here because in this rubber plant, for example, because you're very likely to develop cancer as a result. However, physicians can certainly screen people and advise them that working in a rubber plant may be a very bad idea for them because they are likely to develop cancer as a result. So, and if we look at the reduction in cancer deaths in the United States, over the past 30 years, a few % of that reduction has been due to treatment. And the other 95, 6, 7% of that reduction has been due to prevention. And basically from preventing people having exposure to carcinogens. So if we can find people that are
Exposure to what?
carcinogens.
Oh, okay.
So, and if we can find particular chemicals that people are sensitive to, then we can likely make these people healthier a lot longer. Next slide, please.
So, can I say-
yeah, please.
So these, those outliers, have they looked at like what kinds of metabolic pathways that these, are there certain metabolic pathways that, you know, may have,
Why they're sensitive?
why they're sensitive? Explain why they're sensitive.
Absolutely. So that would be the next step. So the slide before came from some work that I did with a postback at NIH on this SAGE challenge or whatever. And we simply looked at hypersensitivity. If you had more data and more cell lines, which we now have, so we have, there's a repository of about 6,000 human cell lines. So if you had more data and more cell lines, those are certainly things you could look into. And for that particular compound, that's something you could probably look into right now. I have not looked into that at this particular point. But that's, so going back to the example of maple syrup urine disease, that's exactly what they did. So they found a particular mutation and they said, well, what is this? You know, what is the amino, or what is the protein that this thing encodes? Oh, it's involved in the pathway that processes branched chain amino acids. We're going to hypothesize that these kids have a buildup of branched chain amino acids. Sure enough, they did. Yeah. Okay. So this, and by the way, this data you can get is fantastic. This data was made by a guy named Mike Schneider. Mike Schneider is the head of genomics at Stanford University. So Mike Schneider decided that he wanted to sequence somebody a whole bunch of times. And he sequenced himself up to the point of this study 19 times. And the reason he said he sequenced himself is because he was the only person he could be absolutely sure wouldn't sue him. Subsequently, he has sequenced his mother a bunch of times. But anyway, so far she hasn't sued him. And so far, his mother has not sued him. But anyway, so Mike Schneider started sequencing himself. Sequenced himself for these are days, so a couple of years. And what he would do, what he really focused on. What he would do is he would sequence himself to sort of establish a baseline, and then every time he got a cold or a respiratory virus, or when he got a cold and a respiratory virus, he'd sequence himself a lot to see what was going on with the genes that he was expressing. And so he was able to sort of make these profile and see what was going on with himself. And it turns out that after this nasty respiratory virus infection, he ended up with diabetes. And apparently, so it's funny because I work at the National Library of Medicine. We have all these old books, these old wacky books. So I was giving another talk about this subject 6 months ago. And I started looking into diabetes onset after viruses. It turns out in the early to mid 80s, there was an entire literature on diabetes onset after virus. So, I mean, it's something that we certainly knew about, but there wasn't a whole hell of a lot we thought we could do about it. So, but now we can probably start to predict which people have those types of mutations. Let me give you a better example of that. So there's this guy at the University of Washington. His name is Skip Virgin, Herbert Virgin, if you want to look him up. And Skip Virgin started studying mice and people with an autophagy. So autophagy is how cells grab and eat things. And he started studying mice and people with this mutant for some reason, and they had very little phenotype. So they really didn't seem to be abnormal at all. So somehow, one of the people he was studying got persistent norovirus. So this is the kind of thing people get on cruise ships, right? Where they get sick for a bunch of days. Now, I should tell you that there's two kinds of norovirus, two basic kinds of norovirus. One is persistent norovirus, which you get for days, and it's people that have people at home and so on. And then there's transient norovirus, which you know, it's like a little bout of food poisoning. You know, you get sick for 8 hours, it's unpleasant, and then you're done. So what he found, and what is fascinating, and he hasn't published yet, but I'm very hopeful that he will soon, is that both people, and he's confirmed it in mice, if they have this autophagy mutant and they get persistent norovirus, they all come down with irritable bowel syndrome, or the mouse equivalent of irritable bowel syndrome, which I have no idea how they detect that. But anyway, and if they get transient norovirus, they see no phenotype whatsoever. And it seems to be very specific to norovirus. That tells us that there are particular genomic backgrounds where if you get very specific viruses or you're exposed to very specific toxics, then you're very likely to get sick. And in this case, you're very likely to get chronic diseases, which can be a problem for a long time. But we'll talk later about treatment for irritable bowel syndrome and how metagenomics plays into that.
Yeah, so, but isn't there a microbiome, angle
That's
to?
exactly in the context where we're going to talk about it later.
All right,
Give me
okay,
about
okay,
5 slides.
okay.
All right, next slide, please. So, metagenomic and diet affect disease states. So we're going to pretend in our stomach disease X, we've talked about the genomic-only cases, and we've talked about toxics and bacterial and viral infections, and now we're going to talk a little bit about metagenomes. All right, so next slide, please. So we have about 10 trillion human cells in and on our bodies. We have eye cells, and gut cells, and foot cells. We don't have foot cells. Sorry. But anyway, we have about 100 trillion bacterial and fungal cells in and on our bodies. Now, fungal cells we don't think about much because we don't understand fungi very well, and also typically in metagenomic sequencing, we hadn't been sequencing them. And the reason is that their membranes are actually really hard to crack. And it's a delicate balancing act because you want to crack the fungal membranes without degrading the DNA. So we were way undercounting the fungi. And now we know there's probably a lot more fungi than we thought. For example, our foreheads. Our foreheads are a fascinating ecosystem because our foreheads, remember, on our forearms, there's hundreds and hundreds of different species of bacteria, right? And they live here happily, and I'm going to talk about some of those species of bacteria in 4 or 5 slides. But on our foreheads, there's really only one species of bacteria and one species of fungi. And they really outcompete everything else. And that's a really fascinating thing to think about because our foreheads are totally open to the air. There should be bacteria and fungi landing and colonizing there all the time. But they're not because they're outcompeted because the bacteria, Propionibacterium acnes, as well as the fungi, which I have no idea what it's called because nobody studies fungi, but tell me. But anyway, so they are so well adapted to that particular ecological niche that absolutely nothing else can really survive there for very long and appreciable amounts. But anyway, so we have all these bacterial and fungal cells in and on our body. No more asides on that. Next slide. So, and one of the reasons that this might be important is because of antibiotic resistance. So we take lots of antibiotics, they feed, we eat meat, they feed our food, lots and lots of antibiotics. And so we are seeing that antibiotic resistance is coming up. Now, one piece of heavier news here, and one thing I worked on for a while, is making more specific antibiotics. And also making antibiotics that are specific to pathogens and not so much to non-pathogens. And that's I think, the direction where we're going. But there are other problems besides potential generation. Remember, this is a stochastic process, so you don't generate these necessarily in a linear fashion, but at the same time, there are other problems with taking too many antibiotics than just the antibiotic resistance. And we will go over those over the next few slides. However, the one thing I want to say on this slide, I remembered what I was supposed to say, is that, so there's a bacterium which is the #1 nosocomial infection in the United States. So nosocomial infection is something you pick up in the hospital. That's called Clostridium difficile. 90% of Clostridium difficile cases come after antibiotic treatment, typically in the hospital. So what happens is C. diff sits in our intestines and is reasonably well behaved. Typically because the rest of the bugs in our intestines keep it in check. When there are large courses of antibiotics that kill everything, basically we napalm the gut, C. diff has an ecological niche where it can go bonkers. And what it does is, basically there's 2 levels of our gut lining. And C. diff gets through one of the levels of our gut lining, and then our immune system goes nuts. And it attacks C. diff, which makes C. diff go even more nuts, and then we have a major health problem on our hands. Does that make sense to everybody? So that's yet another problem with the overuse of non-specific antibiotics. Next slide. Oh, there's the whole C. diff thing. Okay, so C. diff is also associated with colitis. Colitis is fairly similar to IBS, which I discussed earlier. So, now we have a fairly effective way to treat IBS. And what that is called is fecal transplantation. So, about 6 months ago, I met this meeting of the Institute of Medicine in DC. So this is part of the National Academy of Sciences, and it's basically the government's panel on what we're going to do about certain diseases. Anyway, so I'm sitting there, and it's some sort of metagenomics meeting, and there's all these very serious people. So there's these generals, you know, the people with like all the so on and so forth. What's that? Bars. Yeah, it was the people with lots and lots of bars, so, you know, like this many bars, right? So they're sitting there and they're very serious. Until this guy comes up, and he's from the Netherlands, and he's talking about fecal transplant surgery. And he's got videos of like preparing the poop and stuff. And really, I mean, for one thing, so the amazing thing was that this is 85% effective in this guy's hands. I mean, 85% of people with IBS, which used to be this horribly intractable disease, come out with no more symptoms for a couple of years. The other funny thing about it was that these people with a whole bunch of bars are sitting there asking questions which are thinly veiled poop jokes. I mean, the poop jokes just abounded from these people, from the whole audience for quite a while. So that was really amusing. But anyway, so that's what I have to say about fecal transplantation. And the reason it works is because we're taking gut bacteria from healthy people and we're moving it into people, actually not gut per se. We're taking colon bacteria from healthy people and moving it into people whose colons have become not so healthy. The next couple of slides might explain a little bit why that works. Oh, first. So first I wanted to talk a little bit about the skin bacteria on our forearms. And I'm not here to make any health recommendations, but maybe I'll change your mind a little bit about Purell. That said, I still think Purell should be used in a medical context. If you work in a hospital, please continue to use Purell. If you don't work in a hospital, you might consider the following. Staphylococcus aureus is a nasty bacteria. It is a nasty bacteria that causes nasty skin lesions. Some of those are antibiotic resistant, and we have sort of big public health problems with them. Staphylococcus epidermidis is a related bacteria. Most strains of epidermidis, there are a couple that are pathogenic, but 95% of epidermidis strains are not pathogenic. In fact, they live on our normal skin pretty much all the time. They in fact, on our forearms, they are the predominant species of bacteria. But also, what epidermidis will do is it will actually detach biofilms of Staphylococcus aureus from our skin, and they will fall off and die. So, but it's important to note that Staphylococcus epidermidis and Staphylococcus aureus are exactly as susceptible to Purell. Just a thought. All right, next slide, please. So, I want to talk a little bit about the work of a guy named Jeff Gordon, and this may explain the idea. So, Jeff Gordon is a guy at the University of Washington who works on microbiome stuff. He's sort of I guess, one of the biggest players in microbiome research, particularly gut microbiome research. And he has a bunch of interests, but one of the things he's particularly interested in are twins in food-compromised areas. So places like Bangladesh, Malawi, a couple other countries where kids have maybe enough to eat, but there's some food insecurity. And so he works on twins in those areas, identical twins, where one is severely malnourished and the other kid looks fine. And what he finds, and this diagram really illustrates, is that you have one of the twins who gets diarrhea. They get an issue with having some pathogenic bacteria in their gut, and you start to get a vicious cycle. When there's a little bit of food insecurity, you start to see decreased immune function, and basically the bacteria in their gut change from Bifidobacterium and other nice bacterium that we like to see in our gut. I try not to anthropomorphize too much, to what are particularly nasty bacteria and really associate with, I would say, negative health outcomes. In this case, malnutrition, because these kids aren't getting quite enough. So that's a real issue of concern, but also an issue of hope for places like the developing world, if we might be able to treat these kids with microbial supplements. And also, of course, this plays into the whole probiotic debate, which the jury is still out on. The main people that are putting out probiotics are pet care companies in Britain. So pet care companies in Britain have all kinds of graphs for what they will put into your dog's gut. And they have very fast-growing benign bacteria that will grow in your dog's gut, which is probably not the worst thing, but I mean, there's still some work to be done on that. So I would say we'll know more about probiotics in the next couple of years, and literally a couple of years. But certainly, I tell my friends, and once again, I'm not here to give medical advice, but I think if you were going to try to eat more healthy, less processed, less fatty, and more sort of nutritious food, it might be best, really the most important, to do that after a course of antibiotics. And that's actually not just me talking, that's also Jeff Gordon talking. Because what he did another experiment, which was somewhat unrelated to this. So he takes gnotobiotic mice, so mice that have had their whole digestive system removed to bacteria. And he feeds them either fruit or basically fatty food. And I'm not a vegetarian, but he feeds them either fruit or mostly fatty food in their intestines after feeding them this kind of food when they're released from the gnotobiotic cultures. And it turns out that healthier bacteria grow in the mice that are eating more fruit than fatty types of food. Does that make sense to everybody? I mean, it sort of makes intuitive sense, I think. And now we're starting to see those types of things people are. Next slide. So bacteria can also change the way our human cells respond to things. And that is shown by this slide. And I just want to walk you through it because this is a bit of a scientific slide. So what this is it's two different populations of bacteria. So these are gut epithelia, and they're cultured with two different populations of gut bacteria. And I was taking the train up here, and I said, I wonder if I can remember, just off the top of my head, which populations of bacteria they were, and I don't. But the point is that they're two quite different populations of bacteria. I mean, I think one is an alpha proteobacteria. No, neither of them are proteobacteria. They're typical gut bacteria, but one is a phytobacterium or something. Some sort of enteropathogen. And what you see here is this is gene expression. And you see that the gene expression is pretty much opposite. In the gut epithelia, expressed, these genes are down and these genes are up, and these genes are up and these genes are down in the gut epithelia exposed to one kind of bacteria versus the other. So this gives us a real clear indication. Now this is an in vitro study, so I mean, it's not necessarily as clear cut in the gut, of course, but what we're seeing is that the transcriptional pathways in the gut epithelia are really severely impacted by the bacteria that are around. Next slide. All right. How's everybody doing?
Good.
Everybody okay? Great. All right, so let's talk about epigenomics. We'll move on to that. I'm going to give a little bit more background on epigenomics for the non-biologists in the room. Next slide, please. All right, so if you guys remember the central dogma of molecular biology, DNA goes to RNA, goes to protein. More or less. There's a lot of other stuff that happens. We don't know. There's as many long non-coding RNAs as there are protein coding genes and so on and so forth, but ignoring that, DNA, RNA, protein. Everybody comfortable with that? Great, next slide. Okay. This slide is a little more complicated. So basically, it doesn't look like this typically, actually. Well, it looks kind of like this in bacteria, but in eukaryotes, typically genes are spread out. But this schematic basically shows you'll have a transcription factor, when expressed, made into a protein, will activate different genes. And different genes may or may not activate each other. And then you can have different pathways, which can result in over or under expression of different proteins. Does that make sense? So let me go through that again.
Could you say that again?
Absolutely. So you have transcription factors.
Yeah.
Right? So a gene is expressed, which then is made into a protein, and that protein will bind right before other genes and turn those genes on.
Okay.
And some genes turn other genes on and off, and then remember these genes all become proteins, and those proteins interact with each other. Okay, so yeah. Okay, great. Next slide. All right. And DNA, just to remind you from high school biology, DNA is packaged in a chromatin. So this is DNA. These are histones. And DNA is wrapped around histones, so on and so forth, fibers, chromosomes. So the important thing to remember is that the DNA has to be exposed in order to be transcribed. It can't be wrapped around and packaged into here and so on and so forth. And there's a whole bunch of mechanisms to do that. Next slide, please. So we see that these things are wrapped around the nucleosomes, so on and so forth, and there's linker DNA. So they have to get somewhat unwrapped. Next slide, please. And so, actually, go back to that slide, please. So these histones can be acylated, and they can be methylated by a whole host of factors. We call histone methyltransferases, and histone acylases, and deacylases. And those determine the wrapping of DNA around these particular things. Now the reason that's really important is because these things, so transcription factors probably regulate dozens to hundreds of genes. Histone acetylases and deacetylases regulate things on whole different levels. So they will regulate hundreds to thousands of genes. Does that make sense?
Because they
So
regulate how exposed they are?
What's that?
Because they regulate how exposed they are?
Yes. So yeah, it regulates how exposed the DNA is to be able to, for transcription factors to bind, and then the transcription initiation complex to bind, because those are pretty big proteins too. So they have to get into it. DNA can also be directly methylated, which also changes its transcription. And then there are particular sites, which we know now, on the histones, which either sort of turn genes on or turn genes off. So for example, histone 3 lysine 4 trimethylation tends to turn genes on, and histone 3 lysine 27 trimethylation turns genes off, more or less. And that's way too much detail. So let's go to the next slide. But this is very important because we have about 146 cell types in our human bodies, and this is more or less what determines which cells become what. So obviously, a skeletal muscle cell, which is a microlung, is very different than a red blood cell, for example. And so this is the major mechanism by which our cells differentiate into different things during fetal development, and also maybe a little bit later on. Next slide, please.
Are you saying that the major difference is the substance that wrapped around?
Yeah, the methylation or acetylation state.
Okay.
So there's lysine, which I'm not going to draw. I'm going to resist the urge to draw the chemical structure of lysine because it's way beyond the scope. But anyway, there's these lysines that are chemically modified. And those chemical modifications determine how tightly the DNA is wrapped around those histone proteins, and that determines which cells become what.
And how many variants are there of the lysine?
Well, so there's not a ton of variants of the lysines themselves. I guess technically you can be, let's call it 6.
Okay.
Each lysine, and then there's probably on the order of 12 lysines on, there's probably a total of about 30, 40 lysine residues in the whole histone complex that are relevant to this process.
Okay, so are you saying that there can be 30, 40 different types of cells based on that? Or is it more complicated?
No. So remember, let's go back.
Always more complicated.
So we had, can we go back? Yeah. So we have, remember, we have 3 trillion base pairs of DNA, right? 3 billion, sorry. 3 billion base pairs of DNA. This is 146 base pairs.
Okay.
So basically, there's one of these on every 146 base pairs, and these are specifically modified to release or bind more tightly to the DNA.
Okay, so it's combinations at different points in the DNA.
Exactly, combinations at different points of the DNA that release them, and so on and so forth. But the really important point is that these factors that can do this, when they're turned on, can regulate hundreds or thousands of these things, right? And hundreds or thousands of genes, because there's probably many of these spanning a gene, a single gene.
But then is the DNA coded in some way that targets a specific lysine variant to a specific point on the chromosome in order to produce a certain kind of cell, or is this just sort of random that these things?
It's absolutely not random.
Okay.
So yeah, these things are drawn into the DNA and then they hit specific histones. Yeah.
Okay.
So we can discuss it more offline if you're interested, or I can actually just send you to the head of epigenomics for NCBI who teaches this stuff. Who, by the way, I stole these slides from Ian Fingerman, who is the head of epigenomics at NCBI, so I should thank him right now for stealing those slides.
Ian.
He's a good guy.
What is his name?
Ian Fingerman.
It looks like he stole it from someone else because it says Freeman on the bottom.
Oh yeah, no, we all, so this is the credit.
It's a textbook
he didn't publish
publisher.
this particular electron micrograph, and so yeah, I mean, we all take it. All right, so next slide, please. So that's how we get all these cells. But if these processes go awry, very strange things happen. Next slide, please. Right. So to recap, all the cells are essentially the same DNA. But a cell expresses only a fraction of its genes. So if we do RNA-seq, which we can do now, what we see is in your typical RNA-seq experiment, which is a whole lump of cells at a given particular developmental time point in a person or an animal, you see, for an animal, you see about 1/6 of the genes, 1/6, so 16%, being transcribed at any given time. Does that make sense? Okay. So, and then different types of cells arise and that's mostly blood cells. Different types of cells arise because different sets of genes are being expressed, and the pattern of genes that are expressed can be changed in response to the environment. Does that make sense? Everybody with me? So the things that methylate and acetylate these histones are responsive to the environment. All right, next slide, please. So when gene granulation goes awry, we start to see strange things. We see twins who are very, very different, for example. We see people with extra fingers. We see very strange wing patterns in flies, and we see diseases like chronic myeloid leukemia and rheumatoid arthritis. Not that all rheumatoid arthritis is linked to genetics, but certainly some of it is. And that goes back to this etiological point. Remember, this is only some percentage of any given disease. Sometimes it comes from more than that, particularly if it's cell type differentiation type stuff. Does that make sense? Great. Next slide. But this is fascinating. So remember, I told you environmental toxins can affect these types of processes. So has everybody heard of bisphenol A? Everybody
Yes.
knows what that is? Right. So this is fascinating, is that the mother of this mouse received, so the mothers of both of these mice were exposed to BPA while they were pregnant. The mother of this very strange looking obese mouse was fed a normal diet. The mother of this mouse had a diet supplemented with a whole bunch of methyl donors. So basically what BPA does is it messes with the methyl donors that mediate this epigenetic phenomenon. So methylate histones and DNA, right? We can compensate for that by giving them extra methyl donors. And you see, this is what a normal mouse is supposed to look like. Yes?
Oh, no,
So,
I was just stretching.
what's that?
I was just stretching.
Okay, cool. Yeah, not saying that we should give people, we should expose people to much PPA as they want, as long as they take folic acid and vitamin B12. But, it is fascinating to see that epigenetics can be directly affected by these things. All right, next slide, please. All right, so bringing it all together. This is the one slide for biologists, so speak now if you have questions. So, I think what we're going to see over the next few years are really syndromic disease clusters start to form. And I'll give you a couple examples of what I mean by syndromic disease clusters. I think a simple example is something like GATA2. So GATA2 haploinsufficiency happens to a lot of people. Don't worry, if you're not a scientist, you're not responsible for this slide. So GATA2 haploinsufficiency happens to a lot of people, but the syndromes they get as a result are extremely varied. So some people present with myelodysplasia, other people present with types of leukemias and lymphomas, and other people present with all kinds of other bizarre things. And that says the haploinsufficiency of one particular protein is giving you a bunch of different diseases. A wider spread example, which some of you guys may be fairly familiar with, is inflammation. So inflammation, widespread inflammation, gives people all kinds of strange chronic diseases. A lot of autoimmune diseases, but also things like asthma, things like multiple sclerosis is linked to inflammation. So we see there an underlying cause which gives you a variety of syndromes probably based on your genomic makeup. Does that make sense? All right. Any questions on this one? All right, next slide, please. We're getting there. All right, so bringing it all together. As I said, for most diseases, there's a complex mix of genomic environmental factors. We all sort of knew that intuitively. Next slide. But it is no longer a black box. We're starting to be able to tease out things from genomic and environmental interaction. And we're starting to be able to do great things, like put them into databases so we can actually cross-compare those things. Next slide. And so each time, going back to the genomics part of the talk, remember, each time we discover a genomic driver for 10 patients, 20 patients, 50 patients, whatever it is, we save millions of dollars, not to mention pain and suffering. So it's something really worthwhile from a public health context. The other thing we can do is remember, every time we find a genomic etiology, we can take those patients out, separate them out, and we have a better chance of finding other genomic etiologies for that same disease. Does that make sense? Great. All right. Next slide. And by being aware, and I kind of said this explicitly before, but really being able to do targeted prevention of environmental factors, we really changed the conversation. And we changed the conversation to not preventing death, not giving people 2, 3, 4 more years to live, but giving people 25 more healthy years. And that's something that has a huge impact on both people's lives and sort of public health in general. Next slide. Thank you. So what can you do? Well, has anybody ever seen a Cialis commercial? I don't even know what Cialis is, but talk to your doctor about genomics. Really, I mean, getting clinicians on board is a big thing, and that's why I always hope there's not clinicians. And hopefully they're not watching this on the internet. They're too busy anyway. I would have lost a little bit. So please talk to your doctor about genomics. Tell your friends, and also you can conduct a family health history. That's actually a big thing, especially if at some point you decide you want to have your genome sequenced, which might happen in the next few years. And I promise at the end we'll talk about 23andMe. Okay, so you can conduct a family health history, and HHS, the Department of Health and Human Services, has a whole online setup for you to be able to do that. And you can do that locally. HHS doesn't spy on people like maybe other governmental institutions have been known to do. And that really may be a useful thing to you. Even if you're not interested in genomics, it's probably a good idea for the health of you and your family and your kids and all that stuff. Next slide, please. If you're interested in following up on more of this stuff, I highly recommend the Genetics Hope reference. So this actually has a wealth of excellent information, and it will link to more complex information if you really want to go find that type of stuff, like the genetic testing registry and so on and so forth. So this is a great resource. Please use it. And